Start this interactive lesson right by downloading your Microbiology It workbook. Click the link in the description box and let's microbiology it. The gastrointestinal tract consists of distinct microenvironments: the stomach, small intestine, and large intestine, each differing in acidity, oxygen levels, and microbial density. The stomach is highly acidic and contains relatively few microorganisms. Those species from phyla such as firmicutes, Bacteroidetes, and actinobacteria can survive transiently. The small intestine transitions from an acidic duodenum to a less acidic ileum, where microbial populations become more established. The ileum, in particular, is anoxic and supports a dense community of bacteria capable of attaching to the intestinal wall and fermenting nutrients. The large intestine, or colon, serves as a major fermentation chamber containing the highest bacterial density in the body. Facultative aerobes consume residual oxygen, creating conditions that favor obligate anaerobes such as Clostridium and Bacteroides species. Welcome to the second part of this video where we are going to micro mind map gastral tract. This is the first part. First part, we're going to talk about the environments. We have the stomach, the small intestine, and the large intestine. Now with stomach, we are going to remember acidic and low microbes. With the small intestine, it's anoxic and there is high bacterial density. So that will be our second stem from the small intestine. And our third one would be fusiform anaerobes. Well, on to the large intestine. We have strictly anoxic, and the bacteria that are in the large intestine is found facultative aerobes and obligate anaerobes. And lastly, stemming from the large intestine, in vivo fermentation vessel. The gut microbiota is composed primarily of two bacteria phyla, Bacteroidetes and Firmicutes, which together account for roughly 99% of all microbes in the colon. Minor constituents include archaea, for example, Methanobrevibacter smithii, eukaryotic yeast, and transient protozoa. These microbial groups exhibit metabolic diversity, enabling the host to digest complex plant polymers and extract otherwise inaccessible nutrients. Researchers have classified gut communities into three functional enterotypes: Bacteroides, Prevotella, and Ruminococcus. Each enterotype differs in metabolic capacity, such as protein degradation, carbohydrate fermentation, or mucin breakdown, and may influence an individual's dietary response and drug metabolism. The composition and dominance of specific enterotypes are shaped by long-term diet, genetics, and environment. High protein, high fat diets are typically associated with Bacteroides, while carbohydrate-rich diets favor Prevotella. These microbial networks are not just passive passengers, they interact dynamically with a host's immune and metabolic systems, forming a symbiotic foundation for nutrient absorption, immune development, and gut homeostasis. All right, you're doing great. So, our second branch of our gastro tract micromind map is gut microbial. Stemming from that one, we have Bacteroidetes, gram positive, firmicutes. Our next branch is archaea, Methanogenesis. Our last branch of the gut microbiome is enterotypes. There are three. The first one is Bacteroides, the second one is Prevotella, and the third is Ruminococcus. Stemming from Ruminococcus, we have mucin and polysaccharide. So mucin is degraded and polyside is broken down. With Prevotella, carbohydrate fermentation, and with Bacteroides, protein and fat metabolism. Bacterial fermentation of dietary fibers yields short chain fatty acids such as acetate, propionate, and butyrate, which serve as energy sources for colonocytes and regulators of immune signaling. Microbes also synthesize essential vitamins, including B12 and K, which the host cannot produce independently. Moreover, microbial metabolism affects the enterohepatic circulation of bile acids and amino acids, influence digestion and energy balance. A notable example of symbiotic regulation is Bacteroides fragilis, whose polysaccharide A mediates immune tolerance by promoting regulatory T cell activity and protecting against inflammatory conditions such as colitis. Conversely, pathogenic species such as Heliobacter pylori illustrate the thin line between commensalism and disease. This proteobacterium, transmitted orally among family members, can colonize the stomach lining, leading to ulcers and gastric malignancy. On to our last branch. Our third one is host physiology. We have one, two, three, four stemming from it. The first one is nutrient synthesis. We have vitamins B12 and K. Our second one, we have the metabolic byproducts. Stemming from that, we have plant fermentation and the byproducts, methane, carbon dioxide, and hydrogen. And then our third branch, we have host interactions. Stemming from that, we have gut-brain axis. Microbial metabolites influence the gut-brain axis. And then we have immune tolerance and inflammatory disease. Microbial polysaccharides promote immune tolerance and prevent inflammatory disease. Clinical relevance. Heliobacter pylori is a pathogenic proteobacterium. You soaked all of the information about the gastrointestinal tract and you've completed the interactive micro mind mapping. Now it's time to test your knowledge with a few problems. If you haven't already downloaded your workbook, which has notes, 20 microbiology problems, and answers, then click the link in the description box to download it. Feel free to pause the video and place your answers in the comment section for these problems. If you found this video helpful, then like, comment, and click on the next video.